So Dave, good morning. I'm not going to read your biography. I think everybody's got that if they've read the calendar invitation. Suffice to say that you are one of the leading people in your field and you've been doing some great work, and we're going to hear about some of it today. So I'll just hand straight over to you if that's okay.
Cheers. Thanks very much, Paul. So Aideen and Nicole asked me to give a talk today on kind of a summary, I guess, of my journey to where I am now. So that's what I'll do for the next 25 minutes. And then Daniel Hwang from my group will talk about some research that we're currently doing. So I'm going to begin the tale back in 1999 when I first got serious about research and decided to do a PhD at the Queensland Institute of Medical Research with Nick Martin, who heads up the twins unit there. My co-supervisor, many of you will recognize in the middle there, that's the now Pro-Vice Chancellor of Research, Mark Blows. And also our own Ian Fraser was an associate advisor of mine. And I decided to do my PhD on the genetics of blood cell concentrations because at that time I knew absolutely nothing about genetics and I knew absolutely nothing about immunology or hematology either. So, you know, why not, I guess. And one of the first things that we looked at was the heritability of blood cell concentrations because Nick had been sending blood samples from the twins over to Ian here at the PA and having them measured in terms of a differential and a lymphocyte subsets analysis. And what we showed is that for a lot of these concentrations, the heritability was actually very high, even for the lymphocyte subsets. One of the other projects that I did was back in the old days of linkage analysis. So basically we were trying to find regions of the genome that were likely to contain genes that regulated levels of these blood cells. So unlike today where we do similar sorts of analyses with SNP array panels with hundreds of thousands of markers, back in these days it was done with microsatellites with about 400 markers across the genome. And what we used to do is we would correlate the degree of microsatellite sharing between DZ twins with the their similarity on a trait of interest. So for example, this is a genome scan looking at CD4-CD8 ratio. So that's kind of a measure that was used in kind of looking at the severity of HIV infection. And so we found a hit on chromosome 11. You know, so this region of the genome is likely to contain a gene that affects CD4-CD8 ratio. Unfortunately, we know now that these sorts of analyses are grossly underpowered. So that finding probably represents a type 1 error. Now, I have to admit that I was actually kind of agnostic with respect to the particular phenotypes that I was looking at. My main interest actually was in learning the statistical methods in order to do gene mapping, because I realized that, you know, once you had a good handle on the method, you'd be able to apply the same methodology to any trait of interest. And one of the techniques I learned was something called structural equation modeling, which has really become kind of, you know, my go-to technique for the last 20 years. And what structural equation modeling does is it allows you to model the relationship between variables in terms of a statistical model or a path model. And it's an incredibly flexible technique. So basically any sort of linear statistical model can be written in this framework. So something as simple as a t-test or a linear regression, all the way to really complicated models. So, you know, once you know structural equation modeling, you kind of know the whole of statistics involving linear models. And it's also a great technique because you don't have to use numbers or equations. You can basically write out everything in terms of these path models, which are actually mathematically mathematically complete descriptions of your statistical model. So you can kind of avoid equations altogether. And I like them because they're very intuitive. They're actually based on you know, some very old work back in 1921 by a guy called Sewell Wright, who introduced the method within the context of the genetics of guinea pigs. So after 4.5 years of a very long PhD, I was very lucky to be offered a postdoc position at the University of Oxford. Here's a few photos of Oxford. This is the Radcliffe camera, which is part of the Bodleian Library at the University of Oxford. You can go for a punt on the River Thames, or the River Isis, as they call it in Oxford, because they like to be different and a bit pretentious. Two of the kind of lesser known but very iconic sites in Oxford are the Turf Tavern, which is where everyone kind of goes after their PhD vivas. It's also where former President Bill Clinton allegedly did not inhale, and also where former Prime Minister Bob Hawke set the Guinness World Record for consuming a yard glass of beer in 11 seconds, I think it was. I actually lived on the outskirts of Oxford, up the hill where the Wellcome Trust Centre for Human Genetics was located. And one of the more quirky sites in Oxford is Shark House, because the fellow that built this in the 1970s decided to do a bit of home renovation, and why build a veranda or a porch when you can put a life-size model of a great white shark crashing through your rooftop. So whilst at the University of Oxford, I was very fortunate to work on a number of large-scale projects. The first of these was what's called the International HapMap project. And this was basically a big international effort to map the degree of linkage disequilibrium in the human genome. This is basically the correlation between genetic markers. So if you look at the human genome, so for example, this chromosome here on the right-hand side of the slide, the human genome often has this kind of block-like structure to it, where markers close by to each other are correlated with each other, whereas the markers further away tend not to be correlated. And the reason why this is significant is that if you know the correlation between markers, you don't then have to go out and genotype every single marker in the genome. You can recover most of the information by just genotyping a subset of those markers, and you still get most of the information. So that was kind of the idea behind the HapMap project. And we're very fortunate because we had kind of early access to this at the time, cutting edge data that allowed us to do a lot of cool things and some interesting projects. And I'd like to just highlight one of these that's kind of been lost in the sands of time now. So this is way back in 2004. And we had the idea that if you knew the SNP genotypes of individuals at a particular locus, you might actually be able to impute their genotypes at other loci that you hadn't physically genotyped. So this figure kind of shows the, on the y-axis, how well you do at just guessing a person's genotype at that position on the genome. And this is a section of one of the human chromosomes. And basically the topmost line sort of shows you that even at these untyped markers, you can still recover something like 90 to 100% of the genotypes correctly, which was kind of quite extraordinary at the time. But it's also possible, I think, to be too far ahead of the curve, in that we did this study back in 2003 and 2004, which is about 5 years before GWAS data became commonly available to people in the community. So 5 years later, when some collaborators of ours actually did this imputation a lot more elegantly and with some nice software, our study had basically been forgotten. So I think if you look it up on Google Scholar, it's probably got about 30 citations or something like that, whereas the imputation paper 5 years later has something like 2,000 or something ridiculous. The other big project that I was involved with was the first Wellcome Trust case control study. So this was really the world's first large-scale genome-wide association study, which involved 2,000 cases from 7 different diseases and a set of 3,000 common controls. Now, whilst most of the excitement was with respect to these 7 diseases you can see here on the right-hand side of the slide, there was actually a smaller scan involving these 4 diseases, 1,000 cases for ankylosing spondylitis and these other diseases. And I was actually lucky enough to take the lead in the analysis of this smaller set of studies. And this is where I met Matt Brown for the first time, because he had provided most of the cases for the ankylosing spondylitis genome scan. So for those of you who don't know what ANKSPOND is, it's a progressive autoinflammatory disease characterized by fusion of the vertebrae over time in severe cases. And so we ran the world's first GWAS of ankylosing spondylitis. And here's the big association with HLA-B27 on chromosome 6. But actually what turned out to be the most kind of, I guess, clinically significant finding was this association on chromosome 1 with a SNP in the IL-23R gene. So people in this audience will know this biological pathway way better than me, but IL-23R is a receptor that sits on Th17 lymphocytes. And so when that receptor is stimulated, it causes these cells to secrete IL-17, which is a pro-inflammatory cytokine. And this was a really important finding because up until that point, this pathway had not been implicated in ankylosing spondylitis. So if you go to PubMed and you type Th17 lymphocyte and AS into the engine, there's no articles that'll come up before 2008. So this was a really significant finding. And it was interesting because at the time there was actually an anti-IL-17 agent called secukinumab, which had been developed by the pharmaceutical company Novartis, and it was being used in trials of other autoimmune and inflammatory conditions. And a few years after our study, Novartis actually began trialing the compound in ankylosing spondylitis, and it worked really well. So in 2016, the company got FDA approval, and now secukinumab is on the market for treatment of AS. So it's great to think that our findings played a small role in that story. So in 2007, I took up my first proper academic appointment at the University of Bristol. So for those of you who don't know Bristol, The Clifton Suspension Bridge is probably the city's most famous site. This was designed by England's or Britain's probably most famous engineer, someone by the resonant name of Isambard Kingdom Brunel. The Clifton Suspension Bridge, the Golden Gate Bridge in San Francisco, was actually modeled on the Clifton Suspension Bridge's design. In the foreground of that picture, you can see the Clifton Observatory, and it's really cool if you go in here into this tower. There's actually something called an Atlas Obscura, and what that is it's a darkened room with kind of a kind of a little viewing, almost like a pond. And the Atlas Obscura has these mirrors up the top that reflect a 360-degree view onto that kind of, you know, little viewing pond there. So you actually get a 360-degree view of the whole of Bristol. The other really cool site, I think, in Bristol is the SS Great Britain, another Brunel design. This is actually the first iron-hulled ship with a propeller built in the world, and it used to be able to make the trip across the Atlantic in about 14 days, which was very, very fast at the time. Bristol, of course, being built on the back of both tobacco and the slave trade. So in Bristol, my focus changed a little bit in that I became interested in a lot of these genome-wide association studies were beginning to be published. I was interested in how we could use that information in terms of whether we could kind of translate that into the clinic. And so one of the ideas that I came up with, and clearly others would be were thinking about the same sort of thing at the same time, was this idea of genome-wide polygenic risk scores. So polygenic risk scores are a way of trying to quantify the genetic risk of individuals for particular diseases. So what you do is you take genome-wide significant variants, and for each individual, you kind of sum them together to get an estimate of that person's overall risk. And so we came up with the idea of, well, it's not just the genome-wide significant variants that are, you know, can be used for this task, but there's information across the entirety of the genome. And so our idea was basically, you know, to add up everything across the genome in order to inform risk. And so this was way back in 2008, 2009. And this kind of method was very, very prescient because now with the rise of direct-to-consumer genetics companies where people are sending away to get their genomes done by these companies like 23andMe. And recently there's been a lot of interest in the genetics community about whether we can start to move these things into the clinic. So this is a paper in Nature Genetics from 2 years ago where they're looking at whether it might be time to do that in terms of coronary heart disease. And these guys were actually very, very positive that we could start doing this. And we're trying to compare the degree of polygenic risk to some of the monogenic, the risk confirmed by some of the monogenic mutations. So things like variants responsible for familial hypercholesterolemia. So I got my genome done many years ago, about a decade ago now, and there are certain websites that you can go to where you can upload that information. The website will impute your genotypes, just like we did 15 years ago. And then it will quantify your risk for disease or for traits or whatever you're interested in. And so a couple of months ago, I decided to do this myself, and this is the results for it. And so what you're looking at is kind of the distribution of genetic risk and where I sit in that distribution. So for example, on the left-hand side of the screen, for this particular trait, I'm sitting very low compared to the rest of the population. So I'm down at like the 5% of the lowest scores in terms of my genetics. Whereas this one on the right, I'm sitting at literally 99.9999% in the top of the distribution. And I could tell you that those two traits represent educational attainment and the personality trait of agreeableness. And so your task in the next 5 seconds is to work out which is which.
Can we plead the fifth?
And
either
so
way,
that's the answer. So Paul Clark, next time we're having an argument, just goddamn remember how agreeable I am. So this sort of illustrates a few things. The first is A, I'm very agreeable and not very bright, genetically speaking. B, these sorts of scores typically only explain a few percent of the variance. So something like educational attainment, this score only explains about 2% of the variance. So you can think of it like as I've taken a test for educational attainment that consists of 100 questions, and on the first 2, I've got 0 out of 2 or something like that. And then thirdly, it's a beautiful illustration of what's been referred to by the great epidemiologist Jeffrey Rose as the prevention paradox. And what that means is that most individuals in the population who have a disease are actually not those ones who are high at risk, but actually most of the cases come from the this part of the distribution. So if you want to think of educational attainment or having a PhD as some sort of disease, you know, most of the people in this virtual room are not actually going to be from this part of the distribution. They're going to be from, you know, the bulk of this normal curve, not at the ends. That's the prevention paradox. The other kind of area that I became interested in terms of translational epidemiology was this idea of Mendelian randomization. So this is a method of using genetics to inform on causality in epidemiology. So rather than doing randomized controlled trials where you put randomize a group of people into let's say a smoking and a non-smoking group, and that randomization means that any confounders on average are balanced between the arms of the trials. And so if you compare the two groups in terms of their incidence of lung cancer, you can say that smoking causes lung cancer. Mendelian randomization is basically nature's own randomized control trial, where you rely on the random segregation and assortment of alleles in order to do that. So if you have a gene, a genetic variant that's associated with an exposure that you're interested in, like for smoking, which there are, you can kind of do, you know, your own natural randomized controlled trial and compare the incidence of lung cancer between individuals who have genotypes in those two groups. So in 2013, Matt Brown had a chair free at UQDI and asked me if I wanted to come back to Brisbane and lead a genetics group at the Institute, which I agreed to do. And since I've been there, my research has kind of involved 3 different strands. The first of these strands has been large-scale GWAS, which I'm sure everyone's kind of seen some of those across the years. So one focus has been the genetics of osteoporosis. So one of my postdocs, John Kemp, has really led that work very successfully. And John now has his own NH and MRC investigator grant and also a project grant as well. So his career is really kind of taking off now. The second theme has been statistical methods development and software implementation. So one of the examples of that is some work, again, based on this technique of structural equation modeling that's so flexible. And Nicole Warrington, who used to be a postdoc in my group, led that. She's now an independent group leader on her own now. And she and I developed a method to partition genetic effects into maternal and child effects. And that has formed the basis of a lot of our kind of new research in this area that Daniel's going to be talking about in a few moments. Something else that we've done is some software implementation. So one of these is a web-based utility called LD Hub, which was developed by actually one of my postdocs at Bristol, Chris Zheng. And this is a website where you can upload genome-wide association summary statistics to, and it will calculate the genetic correlation between your trait of interest and literally hundreds of others, complex traits and diseases. So this thing, since it was first put online in about 2017, has attracted over 400 citations now, and has about 1,500 users a month from all across the world. And of course, the third theme is Mendelian randomization. So in particular, we've been interested in using Mendelian randomization to conduct transgenerational studies. So this is the idea that parental phenotypes affect offspring risk of disease and offspring traits and things like that. So there's a theory in epidemiology called developmental origins of health and disease. And the idea there is that in utero factors have long-term effects on offspring risk of disease. So a number of individuals in my group are carrying that idea forward and using Mendelian randomization in very large cohorts to investigate those hypotheses. So this slide is just a slide showing my group members currently. So I have introduced John. Many of you will know already. Daniel will be speaking in just a few moments. Gunalen Moen is working on DOHAD, as is Tom Bond, who has just joined us from Imperial College London. Gung is a PhD student who's also working in the DOHAD area. Shannon will be starting a PhD with our group next semester. She'll be looking at the relationship between maternal genetic factors and psychiatric diseases. Misha is a PhD student with John Kemp. And Caitlin is a Queensland Exeter PhD student who will be joining us in January next year. She's currently based in Exeter. So I'm just going to finish with an acknowledgement slide, acknowledging all the past and present group members, the funders, and my many collaborators around the world. And so I will stop there and I will hand over to Daniel.
You're on mute, Daniel. Unmute yourself.
So does it, so do I successfully share my screen?
Yeah, you just want to put it on presenter mode, Daniel.
Does it work?
Yeah, that looks great.
Cool. Yep, so, so I'm going to talk about some hard science that's currently doing in our group. And it's about the new methods to impute parental genotype using genetic information from their offspring. And this is a project I have worked with Dave in the past 1.5 years. So as Dave just mentioned, one of the core research themes in our group is to use the genetic approaches to challenge the Doherty developmental origin of health and disease hypothesis, which posits that the maternal intrauterine environment during fetal development has significant impacts on an individual's short-term and long-term health. And to do so, we first need to understand if maternal genotype affects offspring health outcome, and then to quantify the maternal genetic effects. And this is a traditional model to calculate the maternal genetic effects on offspring health, and in which you regress the offspring health outcome by the maternal genotype they are associated with maternal exposure or the maternal intrauterine environment. However, there's a flaw in the model that the mother gives half of her genome to the child, and this opens another pathway for maternal genotypes to affect offspring health. So the result from the traditional model can be biased because you don't really know if the maternal genotype is acting through the maternal intrauterine environment or acting through the offspring genotype. So to properly estimate the maternal genetic effects on offspring health through the maternal intrauterine environment, we need to block the other pathway by conditioning the offspring genotype. And what I want to convey here is that to estimate unbiased maternal genetic effects, it requires genotype from mothers and their children. So here comes the problem. There are very, very few large-scale studies with genotypes of mother-offspring pairs. And then it actually requires a huge sample size to detect unbiased maternal genetic effects. For example, it needs around 10,000 mother-offspring pairs to detect a maternal SNP effects that accounts for 0.1% of the variance in an offspring trait. So fortunately, this problem was first overcome by a study from our group. The Warrington et al. Using genotype of mother-offspring pairs from multiple birth cohort, plus an alternative approach to increase the power to identify unbiased maternal genetic effects on offspring birth weight. The thing is, there's another problem. The birth cohorts only allow you to study the maternal genetic effects on offspring birth-related outcomes. If we want to study the maternal effects on adult later life health problems, such as cardiovascular diseases and type 2 diabetes, we need the genotype of mother-offspring pairs in which the offspring are adult who have developed these chronic conditions. And there are very, very few this kind of data available. So here comes the solution from Dave. If we don't have their mother's genotype, why don't we just impute them? So Dave mentioned impute in his presentation several times, and then I'm actually doing this right now in this current project. So I'm going to explain what's impute again. So according to the Longman Business Dictionary, impute is to calculate the value of something which cannot be easily measured in the usual way by giving it a value based on similar thing. In our case, the value of something is the maternal genotypes, and the similar things are offspring genotype. So the idea is to calculate the maternal genotype based on their offspring genotypes. And this is imputation we're doing. And here's an example. So if we have a pair of siblings who have genotype of AA and CC, what would the genotype of their parents be? I'm going to give everyone 5 seconds to do the imputation here. So the only possibility is that both parents have the heterozygous AC genotype. And we can say that given sibling genotypes of AA and CC, the parental genotype probability of AC is 1. So this is actually quite cool because even if we don't have the parents' genetic data, we still know what they are based on their children's genotype. And let's do another exercise here. This time we have siblings who are both homozygous for the A allele. So I'm going to give you 10 seconds here to do the imputation this time. Okay, so I don't know if you get the answer, but it took me weeks to get it. So we know that they cannot be CC, but they could be either AA or AC, and whether it's AA or AC to some extent depends on the allele frequency of A and C. If the A allele is more common in the population, then it's more likely to be AA. So when Dave first introduced this project to me, he asked, "Hey Daniel, how good is your probability?" And then I was like, I did it quite well when I was in high school. And Dave just gave me this book and said, get yourself familiar with the Bayes rule and law of total probabilities. Let's derive the probabilities of parental genotypes. So I did get myself familiar with the probability, and what followed were weeks of formula derivation. And then we got the probabilities of the parental genotypes given all different combinations of offspring sibling genotypes. So here you can see that for our last exercise, when both siblings have genotypes of AA, the probability of their parents having AA is 2p divided by p plus 1, and the probability of AC is 1 minus p divided by p plus 1. And the P is the allele frequency of A. So if A is more common, you can see that the parental genotype is more likely to be AA. However, there's another problem in this model of imputation using sibling pairs. That is, we cannot distinguish whether the genotype we're imputing is mother or father's, unless the genetic variance is on the sex chromosome. Therefore, we constructed another model using half-sibling pairs. And this way, we can impute the genotypes of the shared parents and the genotypes of the non-shared parents. So if the shared parents is the mother, then we know that we are imputing the maternal genotypes and vice versa. So here came another round of formula derivation, and then we also got the probabilities of the shared parental genotype and non-shared parental genotypes, given all combination of half-sibling genotypes. And as a part of the method development, we ran a series of simulations to test whether the maternal genetic effects estimated using imputation are the same as those estimated using non-maternal genotype, the real maternal genotype. So the y-axis here in these box plots is the beta of the genetic effects, and the light green backgrounds are the effects estimating using the known or the real parental genotypes, and the pink background are those estimated using the imputed parental genotypes. So basically, the results show that the estimated effects from imputation lined up really well with the real effects. So this gave us confidence that we are doing it correctly. So we still need to face the power issue. So previously we showed that it needs around 10,000 mother offspring pairs to detect a maternal SNP effects that accounts for 0.1% of the variance in an offspring trait. So if this time we don't have parents genotype using imputation, how many siblings or half-sibling pairs do we need to detect the same maternal SNP effects? And we do a power calculation, and then it shows that it needs genotypes from around 33 sibling pairs to detect the parental genetic effects, or around 52,000 half-sibling pairs to detect maternal or shared parental genetic effects. So basically, it means that we still need a large sample, but having something is better than having nothing. So lastly, so it goes a little bit faster than I thought. As there are millions of genetic variants across the whole genome, it would take forever to do imputation one by one in R. So we developed a software called IMPISH, imputing parental genotypes in siblings and half-siblings. The IMPISH does 3 jobs. First, it identifies siblings and half-sibling pairs from a given genetic data set. Then it imputes the parental genotypes using the formula we derived. Then it runs a linear mixed effects model to calculate the unbiased parental genetic effects. This saves a lot of time for people who want to apply these methods to impute parental genotype in their own data sets. In summary, we developed a new methods to impute parental genotype using offspring sibling and half-sibling pairs that enables the estimation of unbiased parental genetic effects without parental genotypes. And then it comes with the software IMPISH and power calculators. A limitation is that it still needs a large sample to achieve sufficient power. So we recommend using these methods as complement to existing large-scale parents-offspring genetic data for estimating parental genetic effects. And these methods can be applied to existing genetic study, such as the UK Biobank, which has around 15,000 sibling pairs. And also the dizygotic twins from twin cohort are useful resources. Then it comes back to our original goal, which is to use genetic approaches to challenge the doe-head hypothesis. With unbiased maternal genetic effects, we can use them in Mendelian randomization to assess the causal effects of maternal intrauterine environments on offspring health outcome. So this project is mainly driven by Dave with assistance from members of the group and collaborators from the University of Hong Kong. Our manuscript is currently under review, and you can find a preprint on the bioarchive. And the IMPISH software and the power calculators are available on our group's website. And that's all from me.
Daniel, thank you very much. That was terrific. Just fleetingly, I thought I really understood it for the first time. That was great.
Thank you.
I don't know how long that feeling will persist, but no, that's really interesting, really exciting, and thank you for such a clear description of what you're doing. So you're publishing that paper now. What are the challenges for you in how you're going to apply this going forward? What are the next areas that you're going to focus on?
So right now we are actually trying to apply these methods. So we have we're trying to improve the software at the moment, and then we are applying the method to the UK Biobank because there are a lot of, as I mentioned in the last slides, there are sibling pairs in these large biobank, and then we're actually trying to impute the parental genetic effects on different offspring outcomes, and then other members in our group are going to use these in combination with our other family data sets to challenge the door-head hypothesis, as I mentioned.
Okay, and what are the sort of particular traits that you're interested in along with your colleagues that you're going to use this method for?
So myself, I'm actually interested in the maternal nutrition, and I did several tests, and then because I feel like the challenge in the field of nutritional science in genetics, nutrition is actually a very messy phenotype in terms of its genetic structure. So even with current really large genetic data sets, we still detect very few genetic variants that show a strong prediction for our dietary behavior. So right now I'm still thinking how to use the data we have at the moment, and that's something I need to work on.
Great. Oh, thank you. Good luck with that. If anybody has any questions, you can flag them up on the chat in case I don't see you waving at me or anything. Dave, can I come back to you? So I've always thought you were very agreeable, but I didn't realize you were quite so special.
Yes, special's the right word for it, isn't it, Tom? When I told one of my brothers my results, he wanted to know whether there was a bug on the website and whether it had reversed coded them.
Now look, don't take this the wrong way, but does that mean that the genetic basis for agreeableness is questionable? I mean, and I mean, I don't mean that in an insulting way. No, but what I'm trying to hint at is those sort of traits, like agreeableness and things like that, which, you know, like everything else, has some genetic basis. But I mean, to me, they're not just complex traits, they're highly complex traits that are influenced with all sorts of factors, like, you know, which side of the bed you got out in the morning and things like that, environmental factors and things. Do you think it's stretching the application of genetics a bit far to kind of link it with traits like agreeableness?
Well, yes and no. I mean, there's always issues in measuring constructs, but you know, you could also probably make a similar argument with a lot of different diseases as well. Like, you know, how well do we measure psychiatric disease? You know, is all manifestations of schizophrenia, you know, kind of the same? I mean, I think that sure, that is one issue. I think two bigger issues are the extent to which traits are the variance is explained by genetics versus the environment. So for most traits, it's probably around 50%. And so if you're just trying to solely predict individual risk using genetics, you'll never ever be able to explain more than 50% of the variance by definition. So that's problematic. And then of course, there's the other problem that for a lot of these traits, the studies are relatively small in the scheme of things. And so that the variance that you actually explain is only in the order of a couple of percent, which is pretty useless for meaningful kind of prediction.
Right. I suppose also it's those sort of analyses entirely dependent on what the measure of agreeableness is, and that's defined probably by a psychologist or someone. And so whether they're really measuring the right traits, if you see what I mean, that can be linked up with a genetic basis. I have some qualms about that, I would say.
Yeah, absolutely. I mean, there's a huge literature on measurement theory and the philosophy of it. You're quite right.
Yeah. So it could be that the trait that the psychologist is measuring, you know, you could find a trait in theory. I'm just thinking off the top of my head, of course. In theory, you could find a trait that could be really closely defined by genetics, but you're not measuring it, if you see what I mean. The psychologist hasn't identified that trait.
Oh,
Okay.
Can I just add to
Aideen,
that?
yeah.
So I was just going to say, usually in these kind of situations, it's one or two items that they're using in order to define agreeableness in a you know a long questionnaire that's capturing lots of things, and obviously some scales are a lot more reliable and sensitive and specific if they're a little bit more in depth and detail and that kind of thing. So this is kind of coffee table kind of stuff more than really, rather than clinical, if you know what I mean.
Yeah, exactly. Yeah. Any questions? Oh, sorry, Dave.
I'm trying not to take this discussion personally.
It's all around your
agreeable nature.
Look, I had to mute myself because I was weeping with laughter when you put that up. I had to turn my camera off. That was really entertaining. I mean, the other one that was perhaps slightly more troubling was the other measure that you flagged up. I mean, what do you make of that?
Well, it's as I said, it's only 2% of the variance. So you can roughly think of that as if you're measuring someone's, educational attainment, which of course is a proxy for IQ. You can think of taking a test of 100 questions, and this is, the answers to 2 questions on that test.
Yeah. And there must be a statistical variance in that anyway. Okay. Any questions? So Dave, I did admit to my age because I remember the SS Great Britain coming back to Bristol.
Wow.
And I thought it was 1971, but actually I've just looked it up. I think it was 1970. So I do remember, and of course, at those days, it was just like a rusty hulk. There wasn't much to it. And it was all completely rebuilt after it was brought back to Bristol. I'm showing my ancient age.
It's an amazing museum now. It's well worth a visit.
Well, we are subsidised by the Bristol Tourist Board, aren't we? It is wonderful. I visited it probably about 5 or 6 years ago. It was the first time I'd been back since I was a kid. That's quite fun. Okay, any further? Okay. The shark is still there, didn't they? The council tried to make them take it off the roof or something, didn't they?
Yeah, apparently it's when I was preparing this talk, I looked it up again, and it's now an Airbnb, so you can actually stay.
So what I never fully understood is if the mouth of the shark is sticking through the bedroom at the top of the house or not.
I don't think so. So apparently the history of it, and I don't understand this, is that it was built as some anti-Vietnam War protest or something. What the hell that's got to do with anti-war sentiment is beyond me. But yeah, it's got quite an interesting history to it.
It's a great British eccentric.
I've just had a question from Anonymous on what is the current consensus on the DOHAD hypothesis.
Okay, serious question. Thank you.
God, yeah, that's kind of opened a big can of worms. I mean, I think that I mean, God, where do I even start? There is a group of epidemiologists that will kind of swear that DOHAD is a thing, and certainly there's a lot of support from it from the animal model literature and also in observational studies. I guess we're probably a little bit more skeptical, and we're interested in trying to examine the validity of that hypothesis using these new genetic techniques of Mendelian randomization. So, that hasn't really been done before, and that's why that's where we're trying to slot in this space.
Okay. If Anonymous wants to come back on that one, you're welcome to. I didn't know you could post questions anonymously. That's great. As long as they're sensible. Okay. Anything further? Okay. Well, our time's up. Dave, Daniel, I'd like to thank you for making that all so accessible for those of us who are not really experts in this area. I thought that was really, really fantastic. And Daniel, thank you so much. I thought your little quiz was really good. I enjoyed that. I'm not sure I got the right answers, mind you, but it was great. Thank you very much.